Hi, I'm a neuron, a specialized cell in human body. Do you want to hear my story? Being a neuron is not easy. My basic job is to receive and react to various kinds of signals so that we neurons can regulate human behavior. However, there are so many challenges. How can I keep myself alive and regulated? How can I react correctly to extracellular cues and communicate with other cells? I have many specialized structures to help me overcome the challenges. Look, unlike other cells, I have a polarized shape. This is soma, the cell body. It contains nucleus, the place for DNA and RNA synthesis, the ER, endoplasmic reticulum, for protein and lipid synthesis and calcium regulation, the mitochondria, producing ATP to provide energy support, and many other organelles. This longest arm is named axon. It can reach to distant places to keep in touch with other cells. Look at its hand. Its name is the growth cone. This part is highly dynamic and plays an essential role during development. These shorter arms are dendrites. They receive signals from other cells via synapse. Those little apophyses on dendrites are called dendritic spines. They are the unique features for dendrites for synaptic transmission. By the way, here is a more realistic diagram just for reference. Since I believe the cartoon is more lovely than the realistic one, I will continue using it in this video. As a neuron, I need to be flexible, dynamic, and polarized enough to function well. Cytoskeleton, those lines in my body, are doing a great job. The colorful lines represent actin filaments, the polarized and dynamic polymers, which concentrate in the growth cone, dendrites, and beneath the plasma membrane. The red lines are microtubules. They are also highly dynamic polarized polymers. The green lines represent neurofilaments. They are non-polarized polymers which support physical integrity. Since the axon goes a very long distance, how can proteins and other cargoes reach to the distal end? There are two ways. One is through axonal transportation, the anterograde transportation to distal ends and retrograde transportation to the soma. Polarized cytoskeletons, the mitotubules and actin filaments, are the roads for axonal transportation, while motor proteins function as cars. Myosin is the motor protein for actin. It is important in short-distance transportation. For microtubule transportation, kinesin carries cargoes like mitochondria to the distal end with the help of adapter proteins, while dynein is the only protein responsible for retrograde transportation. With the help of accessory proteins, dynein carries trophic factors and signaling molecules back to the soma, and old membrane constitutes for degradation. Though axonal transportation is very useful, sometimes it costs too much time to carry the protein. Here comes the local translation. In the axon, smooth ER grows with the microtubules and forms longitudinal tubules to create a web. There are a few ribosomes on it for protein synthesis. In dendrites, besides local translation, there are also Golgi outputs for lipid and protein processing, sorting, and packing. There is a cell signaling pathway for extracellular signals to reach out to me. Some hydrophobic molecules and small molecules can directly go through my membrane, but for those hydrophilic or big molecules, proteins are needed for transduction. Some of them go through transport proteins, while others do not go directly into the cytosol. These molecules, called ligands, first bound to the receptors on the membrane. Then, their signals are integrated and passed on to the downstream signal transduction pathway. Second messengers like DAG, calcium ion, cyclic AMP are engaged in this process. At last, the signals lead to cellular responses like cytoskeleton reorganization or change in gene expression. Here is an example. When the receptors on my growth cone detect some repulsive cues, it would pass the signal down the cell signaling pathway, which leads to the depolymerization of actin filaments on the growth cone. Then the axon retracts. On the contrary, when met with attractive cues, the effect would be the polymerization of cytoskeleton towards the cues. This is the mechanism for me to find the correct way. Since I am a neuron, I need to respond to stimulation and pass the signal to muscle cells or other neurons. This excitatory feature is achieved by a special kind of change in membrane potential, the action potential. The semi-permeability of plasma membrane is a critical factor in action potential generation. The concentration of ions is different inside and outside of the cell membrane. There are much more sodium ions outside the cell, but more potassium ions inside the cell. These ions cannot diffuse freely, so they need voltage-gated ion channels, the red and green dots on the membrane, to go across the membrane. The red dots represent sodium channels and the green ones represent potassium channels. These channels are sensitive to change in membrane potential. When the neuron is at equilibrium, which means the net flux of ions is zero, there is a resting potential. When there is a stimulation that is large enough to make the membrane potential higher than the threshold, the voltage-gated sodium channels open. Sodium ions flow into my body, thus create a depolarization in membrane potential. When the potential reaches the equilibrium of sodium ions, the conductance of sodium decreases and that of potassium gets higher. Voltage-gated potassium channels open. Potassium ions flow out, which leads to a repolarization of membrane potential. The membrane potential keeps getting lower until reaching the equilibrium of potassium, then goes back to the resting potential. This process is called hyperpolarization. After a short refractory period, the action potential propagates down the axon and causes the activation and inactivation of downstream sodium and potassium channels. In myelinated axons, action potential jumps over the strong cells and appears at the nodes of Ranvier. The mechanism accelerates the propagation of action potential through long distances. When it reaches the presynaptic membrane at the end of the axon, it is transformed into chemical signals and causes either excitatory or inhibitory reaction on the postsynaptic membrane. As a result, signals received by the sensory neurons can be passed through the axons. The central nervous system receives the signal and controls behavior by passing on signals through motor neurons. This is how my friend and I control human behavior. Thanks for watching. May the healthy nervous system be with you.